And now, in this chapter, we take a tour of the cell. So our understanding of nature often goes hand in hand with the invention and refinement of instruments. The instruments help to extend our senses. For example, microscopes allow us to see many of the organelles in a cell that we couldn't see with our naked eye. The first crude microscope to observe and make a meaningful record of biological cell was by Robert Hooke, who in 1665 used a crude microscope to observe the bark of an oak tree. And under the oak tree, he observed structures which he took drawings of, the famous drawings, and he termed little rooms cellulae in Latin. And since then, the term cell have stuck with humans. His associate, Leeuwenhoek, then used even more refined lenses to describe living cells. He studied blood cells, sperm cells, and the cells of various organisms that live in ponds, water, where he would take samples of the pond water and observe under the microscope. He even observed living organisms inside his own mouth, his oral cavity. Since the days of Hooke and Leeuwenhoek, improved microscopes and techniques have vastly expanded our view of the cell and will continue to do so for the foreseeable future. As these technologies and inventions take us deeper and deeper into the interior of the cell that we couldn't see before, we now see them at the macroscopic level. We're now seeing them at the nanoscale level and seeing actually how they function even at those very, very, very low scales. So with the advances of invention and technology, we're now able to see the cell. Only in the last 50 years or so are we able to see clearly that the cell is not just a cell membrane and nucleus and cytoplasm, but rather that in addition to all of those and the larger organelles that were originally visible with the help of the high-performing light microscopes, can now, in addition to all of those, we know that the cell has its own skeleton, which is called cytoskeleton. So the cyto is anything of the cell, and the skeleton of the cell, giving the cell its integrity, allowing mobility within the cell, forming similar structure to the human, that the human skeleton and the animal skeletons do for other animals, is present in cells. Members of the cytoskeleton include microfilaments, intermediate filaments, and microfilaments. So this green, pink, and all this colorish network, mesh, like spongy parts of the cytoskeleton. And of course, obviously, the nucleus is the blue blob, prominent blob that we are used to seeing under the microscope when we look at living cells that are not undergoing cell division. So in this chapter, we're discussing the big ideas. We look at introduction to the cell. We take a tour of the nucleus and ribosomes and travel through the endomembrane system. We study the energy converting organelles, their structure, and the cytoskeleton and cell surface. So throughout the cell, we will see how the structure and the function align organisms. So introduction to the cell. The light microscope can display living cells, and with the aid of the light microscope, we studied a lot of what we know about a cell. The way in which the light microscope helps us visualize things that the regular, ordinary eye cannot visualize is through magnification, making the organelle or the cell much, much larger than what the eyes could see. So the greater magnification and resolution of the light microscope compared to our eyes has enabled us to study the cell for many more years. Following the light microscope, which uses a beam of light to magnify the image under the microscope, there is the electron microscope, which uses a beam of electrons to magnify and resolve cells and organelles inside cells. So, the advent of the scanning and electron microscope has helped us to reveal even more detailed structure of the cell, which we now call the ultrastructure of cells. So what is magnification? Magnification refers to the increase in an object size compared to its actual size. And sometimes when you are walking around in the sunny environment, the light passes through you, you might give an image, your image on the floor that will be much larger than your size. Resolution is a measure of the clarity of an image that is generated. So in other words, the ability of an instrument to show two nearby objects are separate. Sometimes you might look at an object under the light microscope and think that it is one, but once you go to a higher resolution of the electron microscope, you will realize that you're actually looking at two different objects as opposed to one. Then you see the layers of cells as opposed to just seeing them thinking that there is one. So you have a higher resolution. In the 1800s, early studies of the cell led to the cell theory. And the cell theory states that all living things are composed of cells, and that all cells come from other previously existing cells. So we've not been able to create cells, but cells are derived from previously existing cells. So back to your checkpoint questions, remember for chapter 4, to collate your answers to your checkpoint questions and email to me. So which type of microscope would you use to study changes in shape of a living human white blood cell? The finest details of the surface texture of human hair, or the detailed structure of an organ in a living cell? So this is paramecium. This image is under the light microscope. You can see the hairs all around it called the cilia, singular cilium. LM stands for light microscope, and it's been magnified 230 times. Here you can see the contractile vacuole. So when it undulates, these spins help to pick up excess water that flows into the animal and is pumped out through the growth. This is the same organism, paramecium, this slipper-shaped animal that lives in ponds around us. It's been magnified 580 times, and scanning electron microscope SEM image. It's been colorized. Electron microscope do not give colored images, but through technology we can differentially treat the surfaces to give different colors. So this is a paramecium. The cilia here is all over the surface, enables it to move around in the pond water where it lives. So this is an internal parasite, and this is a transmission electron microscope. A section has been taken through the cell, and it's been magnified 9,140 times to obtain this image. Again, this is paramecium, so there are now different kinds of light microscope. This is a phase contrast microscope that allows you to see depths and a more 3D-like appearance of a cell under a light microscope. So the phase contrast, what is the composition of the different layers or different organelles allows you to phase the view so that you have a more 3D view of the cell. So this is paramecium again under the light microscope, magnified 380 times, but using phase contrast lenses. Scientists also treat cells with stains and coloration to be able to distinguish the different organs under the various microscopes. In the case of electron microscope, might be treated with heavy metals, and the heavy metals absorbed at different amount where different organelles allow to see them as different colors under the microscope. It is always not very easy to even visualize what it means when we give the sizes of organisms at the microscopic level compared to the electron level. So let's use this chart to visualize what you can see with your unaided eye, what you can see with the light microscope, and what you see with the electron microscope. Objects that are in the order of millimeters up, we can see with unaided eye. So the human egg, so one of the smallest organisms that we can see with the human eye. Most cells you cannot see with the ordinary eye. Exceptions include like the chicken egg, which is just one cell, and muscle cell and nerve cells, which are very, very long. But most of the majority of living cells are microscopic, and the one micron is 1 millimeter divided by 1,000. 1 millimeter is 1 meter divided by 1,000. And some organelles, such as the mitochondria, can be visualized under the light microscope. The whole cell can be visualized under the light microscope, but the outer structure of the cell, including ribosomes and all of the smaller components, cannot, because they are organisms that are sized at the nanoscale level. 1 nanometer is 1 meter divided by 10 to the power minus 9. So it's divided into 1,000 million places. So very, very small. So with a light microscope, most of the plant and animal cells are visible. Prominent in those cells, you can usually see the nucleus of the cell. Bacteria, you can see. Mitochondria also, you can see. But all the other little organelles, they are at the nanoscale level. And electron microscope will be necessary to see viruses, ribosomes, proteins, lipids, and smaller molecules. You require even more powerful technologies and tools to see. Typically, most cells, with the exception of cells like the egg, nerve cells, they are usually small in size. And this relates to the need to exchange materials across their plasma membranes with their surrounding, be it neuron cells or the chemical and physical surrounding. The microscopic size of most cells is important because it provides a large surface to volume ratio. So the plasma membrane, which is a phospholipid bilayer that's embedded with proteins to allow transportation in and out of the cell, it is the surface that connects the interior of the cell with the surrounding. So some protein channels, sometimes we call them tunnels, they shield ions and other hydrophilic molecules as they pass through the hydrophobic center of the membrane. If you recall from the previous chapter, we said the biological membrane, the plasma membrane, are made of phospholipids. In an aquatic environment, the hydrophilic heads turn towards the aquatic environment, and the hydrophobic tails go to the center. So the center of the membrane is hydrophobic, and it's not very, very friendly to hydrophilic molecules. So the hydrophobic layer is not continuous, rather it's injected with, it's sandwiched in between them are proteins that allow hydrophilic molecules to travel through. In addition to the protein channels, there are other proteins that serve as pumps, using energy to actively transport materials in and out of the cell that otherwise cannot go through the biological membrane, the plasma membrane. So compare this cube that is not subdivided to this. The total volume is the same for both, but the total surface area is 54 units squared here, 162 units squared here. So that the surface to volume ratio here is 2, the surface to volume ratio is 6. So there's a lot more surface for interaction, for absorption and excretion into these cells when they're much more smaller. And so it's a more effective in terms of nutrient distribution, chemical distribution, and toxin and waste removal and compartmentalization. So checkpoint question, please remember to answer and email the questions to me. To convince yourself that a small cell has a greater surface area relative to volume than a larger cell, compare the surface to volume ratio of the larger cube and one of the small cubes in 4.2. So this is your plasma membrane, typical illustration. Outside of the cell is an aqueous environment. Inside of the cell is aqueous environment. These are the phosphates, part of the phospholipids. They spontaneously move towards the water, causing the hydrophobic tail, as the carboxylic acid tails, to move into the middle. And as a result, you have 1, 2 layers called a bilayer of phospholipid. In a cell, this bilayer has protein channels. This area is hydrophobic, so hydrophilic materials can go in through these channels. The areas, there are also hydrophilic proteins that are close to the watery aqueous area in the interior of the cell. And the hydrophobic protein regions, they are wrapped by the hydrophobic part of the membrane. So some of the protein channels typically move materials not by active transport, but we also have proteins that actively transport materials across the membrane. So hydrophobic tails, hydrophilic heads. This is a phospholipid, phosphate head, and lipid, diglyceride, and lipid. Prokaryotic cells are structurally simpler than eukaryotic cells. So pro means before, carry-on of the nucleus. So prokaryotic cells are those cells that evolve before the evolution of true nucleus. U means true, carry-on or karyotic has true nucleus. So prokaryotes do not have a true nucleus that's membrane bound. Eukaryotes have all of their DNA material packaged in the nucleus that's surrounded by the nuclear membrane. So they're true nucleus. U means true. All cells have a plasma membrane on their outside. All cells have DNA. They have ribosomes for making proteins, and they have cytosol. So cytosol describes the jelly-like material in the cell. Prokaryotic organisms, the entire interior of their cell is the cytosol. But in eukaryotic organisms, the nucleus is separated from the cytoplasm. So the nucleus has its own nucleoplasm, And the cytosol, or the cytoplasm, is the region between the cell membrane and the nuclear membrane in eukaryotic cells. Bacteria and archaea are prokaryotic cells. All other forms of life on Earth are eukaryotic cells. Eukaryotic cells can be identified, or they are distinguished by having a membrane-bound nucleus and membrane-bound enclosed organelles that perform the same, that perform specific functions. So they're specifically, this is where we learn about how structure and function, the emerging properties as molecules are organized into forming organelles for performing specific functions inside the cell. Prokaryotic cells in general are smaller than eukaryotic cells and simpler in structure than eukaryotic cells. Their DNA is simply wound with proteins, and they are in the cytosol. They are not in their own compartment, in their own membranous house. So these are bacterial cells. This is showing Helicobacter pylori. This is the bacterium that causes stomach ulcers, and they have fimbriae all around the cell, and they have ribosomes. This is their nucleoid, which is the bacterial chromosome, which has DNA and proteins, but no plasma membrane, no biological membrane or nuclear membrane. This is the plasma membrane of the cell itself, surrounded by a cell wall. In some organisms, to protect themselves from digestion by the host cells, or they have capsules. So capsules sometimes help them to recognize and form a colony together with other similar organisms, other similar prokaryotes. So this is a typical rod-shaped bacterium. Eukaryotic cells, on the other hand, they are partitioned into functional compartments. These functional compartments, they are encased in their own membrane. So membrane enclosed organelles, they compartmentalize the cell's activities, and so simultaneously different events can be taking place inside the cell of a eukaryotic organisms. And a particularly disruptive experiment might be taking place in compartment in such a way that it doesn't damage the whole of the cell, and the content of that compartment will be modified and made safe before it's released or allowed to mix with the rest of the cell. The organelles and other structures of eukaryotic cells can be organized into 4 basic functional groups. Group 1, the nucleus and the cytoplasm, they work together to carry on genetic control of the cell. The nucleus is the control center of the cell. It gives a set of instructions, and those instructions are implemented or carried out by the ribosomes. Organelles involved in the manufacture, distribution, and breakdown of molecules, they include the endoplasmic reticulum, Golgi apparatus, lysosomes, vacuoles, and peroxisomes. Then the third group, mitochondria and chloroplasts. These are the cells, these are the organelles in cells that function in energy processing. Mitochondria converts food products into the type of energy that cells use called ATP, adenosine triphosphate. Chloroplasts found only in plant cells help to tap energy of the sun and turn it into molecular energy in glucose and food products after that. Mitochondria is present in all cell types of the eukaryotic organisms, but it's present in all cells, but chloroplast is found only in plant cells. The fourth group provides structural support, enable movement and communication between cells. And these are members of the cytoskeleton, the plasma membrane, and in plants it also includes the cell walls of plants. Remember to email me the compiled checkpoint questions for this chapter. Identify the structures in the plant cell that are not present in the animal cell. So this is a typical cell. The typical cell essentially is used to illustrate all of the possible organelles that we have the scientists have recognized, identified so far to date that is present in the cell. So there's not any cell in which you're going to find only 1, 2, 3 mitochondria. No, or few ribosomes. You have hundreds of ribosomes in the cell. Depending on the activity, energy activity of the cell, you have lots of mitochondria in the cell. So this is a representative cell, illustrative cell. So notice, be able to label all of the parts of the cell. This is the nucleus. Notice that it has its own envelope. It has nucleus. Chromatin is where the DNA is located. DNA wound around histones that give it a three-dimensional structure from the chromatin. This is raw endoplasmic reticulum. It has ribosomes on its surface. The ribosomes also that are freely floating in the cytoplasm. Peroxisomes help to detoxify peroxides in the cell. Golgi apparatus. Notice the flat membrane, almost like pancakes. Lysosomes, they contain lytic enzymes. Lytic enzymes help to lyse cells. Lysis means to break into pieces, break them and digest. Mitochondria, also known as the powerhouse of the cell, converts all the chemical forms of energy to ATP. ATP, adenosine triphosphate, is the energy currency of the cell. It's the molecular form in which the cell can utilize for energy. Energy molecules in all other forms are not utilized. Centrosomes with pair of centrioles. Centrioles are typically found in animals but not plant cells. The animal cells also have their cytoskeleton. Members of the cytoskeleton include microtubules, microfilaments, and intermediate filaments. And all of these organelles and cytosol in the cell are kept separated from the surrounding environment by the plasma membrane. This is a plant cell. The plant cell have essentially the same organelles as the animal cells, with the exception of the centrosome. It doesn't have that. But the plant cells have chloroplasts, which is not found in animal cells. Typical plant cells also have large central vacuoles that gives them buoyancy and turgidity, so that it can grow erect and robust. Sometimes it's packed with chemical material, stored food, and all of that, depending on which part of the plant it's in. The plant cells also have a cell wall made of cellulose, because we have so many plants on Earth, and cellulose the cell walls made of cellulose. Cellulose is the most abundant polysaccharide. Yeah. Along the walls of adjacent cells, there are plasmodesmata junctions that allows the cells to communicate and exchange materials. So because of the plasmodesma, singular, plural plasmodesmata, plant cells' content can actually flow from one cell to another in the phenomenon called cytoplasmic stream. The chloroplasts are important. Notice the green color. They give the green coloration to the leaves of plants. They are the cells that actually are able to tap sunlight energy, giving the plant that functional purpose on Earth to supply energy to other living organisms, and also to supply food or energy as it has, that is harvested from the sun and make it available to other living organisms. Now let's take a look at each group. First, we look at the nucleus and ribosomes in a little more detail. The nucleus contains the genetic instructions for the cell. So because the nucleus houses the cell's DNA, and the DNA directs all activities of the cell, it does so by directing protein synthesis. The DNA is the only self-replicating molecule, and so it can make copies of its strand via messenger RNA. The mRNA can leave the nucleus and go into the cytoplasm and be the blueprint guiding the making of protein, the polypeptide chain. Inside the nucleus is also the region called nucleolus, meaning little nucleus. Because when you look under the light microscope at the cell, the nucleus appears denser than the rest of the cytoplasm. Also, when you look at the nucleolus appears denser than the rest of the nucleoplasm. So inside this little nucleus is where RNA, ribosomal RNA are made, and ribosomal RNA and ribosomal proteins are arranged into subunits of ribosomes. And the subunits of ribosomes, just like the messenger RNA, can leave the nucleus through the nuclear pores and go into the cytoplasm. In the cytoplasm, the ribosomes function in the actual making of protein. So, nucleus works hand in hand with the RNA molecules to make proteins. There are three types of RNA molecules. They're all manufactured in the nucleus. Messenger RNA, which travels independently from the nucleus into the cytoplasm, carrying the message of instructions that will guide the sequence of amino acids that will make a particular protein. Transfer RNA are made, and they are structurally shaped uniquely to go into the cytoplasm as carriers to transport amino acids to the ribosomes during the making of proteins. In the nucleolus inside the nucleus, ribosomal RNAs are used to make ribosomal subunits. These subunits again also go into the cytoplasm. So inside the cytoplasm, the messenger RNA working with the ribosomal RNA and the transfer RNA actually carry out the dream, the function that the nucleus have asked them to carry out. They make the specific protein as directed by the nucleus. Checkpoint questionnaire describes the process that occurs in the nucleus. Remember to compile checkpoint questions and you need to. So this is the nucleus. Here it's extracted and enlarged. This portion is superimposed by an actual transmission electron micrograph, magnified 6,500 times. So these are illustrations to kind of help you visualize what might not be seen as clearly under the microscope to illustrate for illustrative purposes. So this is the nucleus. This denser area is the nucleolus. So this is where the making of ribosomal subunits occur. The subunits will be able to leave the nucleus through the nuclear pore and go into the cytoplasm. All of this is a chromatin. This is where the DNA and the ribosome and the proteins, histones, that help to shape them and fit them into this very, very small compartment. If the DNA in the cell were not wound with proteins to give this globular structure, they will be extremely long and very long. So each nucleus has its chromatin, its chromosomal material enclosed in the nuclear envelope. The nuclear envelope and the plasma membrane is made of phospholipid bilayer. This is one phospholipid bilayer. This is another phospholipid bilayer. So you have a double layer membrane here enveloping the DNA material of the cell. Notice that the nuclear membrane is continuous with the endoplasmic reticulum, and the endoplasmic reticulum has its own interior compartment. Inside this compartment, different experiments might take place, different reactions take place that is compartmentalized away from the rest of the cytoplasm. The ribosomes on the surface of the endoplasmic reticulum give it a rough appearance. So we have two types of ERs. One is called smooth, one is called rough. But on this, the ribosomes on this surface synthesize proteins that go inside the lumen of the endoplasmic reticulum for further processing. So, but let's recap parts of the nucleus: chromatin, nucleolus, nuclear envelope, nuclear pore. The ribosomes, they are composed of ribosomal RNA and proteins, and they synthesize proteins according to directions of the DNA. DNA does the direction by guiding the ribosomal RNA content, the messenger RNA content, and the transfer RNA content that all go together into the ribosomes inside the cytoplasm to make proteins. So when you observe under the microscope cells that are making a lot of protein, you will find large numbers of ribosomes in them. So this is when the ribosomes can be found bound to the endoplasmic reticulum, making it rough ER, or they can be found floating freely in the cytoplasm. If we take one ribosome and enlarge it, you will notice that each ribosome have two subunits. This is a large ribosomal subunit. This is a small ribosomal subunit. Each subunit is made of RNA and proteins. During the manufacturing of protein, messenger RNA is sandwiched between the large subunit and the small subunit, and they read the sequence, the nucleotide sequence on the mRNA. And with the help of transfer RNA, they translate that to an amino acid. And one by one, the beads of amino acids are joined together to form the polypeptide chain, which is the primary structure of protein. Now let's take a look at the endomembrane system. So many organelles are connected in the endomembrane system. So many of the membranes within the eukaryotic cells constitute part of the endomembrane system. They function in the cell for synthesis of materials, distribution of materials, mostly lipids, proteins, phospholipids, and so on, storage and export of molecules. So functions of the organelles of the endomembrane system include synthesis, distribution, storage, and export of molecules. The ER, endoplasmic reticulum, is a biosynthetic workshop. It's part of the endomembrane system. There are two types. Smooth ER does not have ribosomes on it, or rough ER has ribosomes on it. The smooth ER helps to synthesize lipids and process toxins. The rough ER helps to synthesize membranes, biological membrane materials, and the ribosomes on its surface help to make secretory proteins and membrane proteins, which are further processed by the rough ER. Checkpoint question: explain why we say that the endoplasmic reticulum is a synthetic workshop. So the smooth ER is busy making lipids, steroids, all of that kind of lipids. Lipids that will actually be added will be phosphorylated to make the membrane material. So typically, the ER kind of grow in place. They actually make their own components and incorporate and grow in place as the cell grows. So members of the membrane system, this is smooth here. Appear smooth because there's no ribosomes on it. This is rough here with ribosomes on it. These guys make lipids, and these guys make help to process proteins. These guys make proteins, and in here there's a making of proteins and the membrane materials. So the communication, this is how the members of the endomembrane system interconnect. Proteins made in the ribosome, the polypeptide chain grows into the lumen of the endoplasmic reticulum. Inside the endoplasmic reticulum, the polypeptide chain is processed. Carbohydrate fragments are added to make glycoproteins. So the glycoproteins are boarded up in membrane vesicles, transport vesicles, and they get burst off. And the secretory proteins go inside the transport vesicles. They can go into the Golgi apparatus, where they will be processed for export out of the cell or for use in other parts of the cell. Golgi apparatus is also a member of the endomembrane system. It modifies, sorts, and ships cell products. It is connected to the endoplasmic reticulum via the vesicles that move from the endoplasmic reticulum and carry and come back to fuse with Golgi apparatus. The phospholipid membrane being the same for all of these organisms, it's convenient for them to border from one and fuse to the other. So the Golgi apparatus is not continuous with one another, rather it consists of stacks of sac in which the products of the ER are processed further and then sent to other organelles or to the surface of the cell for export. So there's a receiving end. This is a trans end of the receiving end of the Golgi apparatus. And the membrane, vesicle membrane, fuses with the membrane at the receiving end. The content are then transferred through the stack of Golgi apparatus, and while at it is being processed all along, and on the eventually finished product will form a vesicle from the transport vesicle from the Golgi and will be sent out of the cell or another part, a region within the cell. So what is the relationship of the Golgi apparatus to the ER in a protein secreting cell? Lysosomes, they house enzymes, and the enzymes in them are much more like the digestive enzymes inside your stomach. They break down ingested substances and damaged organelles. By making the lysosomes a membrane-bound separate organelle, their toxic content does not mix with the rest of the organelles inside the cell where it could cause damage. It can isolate and break down unwanted cells. In smaller organisms, they serve as digestive enzymes. They fuse with the food vacuoles and they help to digest the food. So how is the lysosome like a recycling center? So in larger organisms, like you have organelles also that are too old. Instead of the cell trying to make itself feed or repair it, it's simply digesting, releasing the nutrients, the molecules that need to be reused again by the cell. In microorganisms, they form colonies, and some of their old colonies will be totally digested by lysosomes and become available food and substrate for the other younger organisms that are growing on top of them. So lysosomes enables the recycling of the atoms of elements that are essential for growth. So in protists. You have digestive enzymes inside the lysosome. This is food vacuole forming as the protist engulfs food from its environment, and the digestive enzyme is released, breaks down the food into useful nutrients. The digestion is completed, nutrients are absorbed by this cell, and you can use it to grow. In recycling, there's a typical cell has a damaged mitochondria. You don't want to leave it there so that it can be infected and cause more trouble. So a vesicle forms around it. Lysosome comes, fuses with the vesicle, digests the mitochondrion, and its molecules are released as useful instead of being destructive into the cell for use. So let's listen to this video on lysosome formation.
Proteins destined for lysosomes are made on ribosomes bound to the lumpy ER and move through the endomembrane system. In this case, the lysosomal protein-containing vesicle that buds from the Golgi apparatus is the lysosome itself. Like secretory proteins and other proteins, proteins destined for lysosomes are made on ribosomes bound to the LUF-ER and move through the endomembrane system. In this case, the lysosomal protein-containing vesicle that buds from the Golgi apparatus is the lysosome itself.
Vacuoles function in the general maintenance of the surface. Vacuoles are large vesicles and they have a variety of functions. In some protists, they have contractile vacuoles because protists live in pond water, and the pond water can rush into their cells, causing them to burst. But because they have contractile vacuoles that continuously pump the water out, the cells maintain their integrity and they don't burst. In plant cells, there are typical large central vacuole that is used to store molecules and waste, and also to facilitate growth of the plant. So checkpoint question, remember to compile and email the answers to me. Is a food vacuole part of the endomembrane system? So this is paramecium and its contractile vacuole. This is central vacuole in a plant. And let's watch paramecium at work, as paramecium's contractile vacuole at work, pumping water. The water is continuously pumped out. So let's review the structures involved in the manufacturing and breakdown of the cell. The organelles of the endomembrane system, they are interconnected structurally and functionally. And in the next table, we can see the relationship among the major functions of the membrane system. So this is the nucleus. The membrane of the nucleus is continuous with the ER. The endoplasmic reticulum can be smooth or rough. The smooth ER is involved in synthesis and processing of lipid. The rough ER is involved in protein synthesis, processing, and the membrane, biological membrane synthesis. And these guys also function in the synthesis of materials that help detoxify cells. This is transport at the end of processing of the synthesized material. They are packaged into transport vesicles, which go on to fuse with the membranes of the Golgi apparatus, releasing its content. The Golgi apparatus finishes processing and packaging of the material. It could be adding phosphor, it could just be processing it for the final function and location. So the synthesized material travels through the Golgi apparatus. It can go into transport vesicle. If it's bound for export out of the cell, the membrane fuses with the plasma membrane, and the content is released to the neighboring cell. If it's bound for use inside the same cell, such as the creation of this lysosome, then the vesicle bolts off into the cytoplasm of the cell. So remember, checkpoint question to email it how to transport vesicles are tied to the endomembrane system. So peroxisomes, these are metabolic compartments that are found in the cell, but they do not originate from the endomembrane system. So remember that peroxisomes do not originate from the endomembrane system. They contain enzymes that can digest peroxides. Peroxides come as a product of digestion of fat in the body, and like we use hydrogen peroxide for sterilization, accumulation of peroxides in our cells could be toxic. But with the help of peroxisomes, those peroxides are converted to water and oxygen and detoxified in that process. Now let's take a look at the energy converting organelles. The energy converting organelles are mitochondria and chloroplasts. Mitochondria, they are organelles that carry out cellular respiration. They are found in nearly all eukaryotic organisms. They have two internal compartments. So they have the intermembrane space between the outer membrane and the inner membrane. And inside the inner membrane is a mitochondrial matrix containing mitochondrial DNA and ribosomes, and as well as many enzymes. In that way, it resembles a prokaryotic cell. The matrix contains the enzymes, some of these enzymes that is used for cellular respiration. Checkpoint question, remember to compile and email them to me. What is cellular respiration? So let's take a look at the structure of the mitochondria. So structurally, because of all the function that it has to perform through a series of steps of experiments, generating ATP from glucose, it needs to be complex, and it is. It's an outer membrane, it has an inner membrane. The space between the outer membrane and the inner membrane is called the intermembrane space. The soup or the matrix of enzymes, DNA, and ribosomes, they are contained in the mitochondrial matrix. So they are all inside the inner membrane. And the inner membrane is not smooth to increase surface area for reactions of cellular respiration. They fold into cristae. So the inner membrane is folded into cristae. To be able to label the parts of mitochondria. In plants, there is chloroplast, and chloroplasts convert solar energy to chemical energy. Photosynthesis is a conversion of light energy from the sun to chemical energy of sugar molecules, and that factory that performs that job in cells are the chloroplasts. So chloroplasts are the photosynthesizing organelles of plants and algae. So this is the chloroplast. Also, given the complexity of the work of photosynthesis, it is outer membrane and inner membrane. Inside the inner membrane is filled with stroma. In addition, there are thylakoid membranes, which are formed into stacks of granum, like a stack of coin. So this is a transmission electron micrograph showing the stack of grain, 3 stacks of grain. So this is should be able to label the parts of chloroplast. So checkpoint question, remember to compile and email them to me. Which membrane in a chloroplast appears to be photosynthetic? Why might it be so? So there is an evolutionary connection. Mitochondria and chloroplasts are are thought to evolve by endosymbiosis. The endosymbiotic theory states that mitochondria and chloroplasts were formerly small prokaryotes that began living in larger cells. So symbiosis is where there's a relationship between two different species of living organisms that is mutually beneficial. Endo means inside. So by the larger organisms engulfing these small protist-like organisms, they get a house to protect themselves. In the case of mitochondria, it helps that cell to obtain energy in the form of ATP. In the case of chloroplast, it helps that cell to tap energy of the sun. And the chloroplast helps to make oxygen available for respiration. So remember, checkpoint compile and email them to me. All eukaryotes have mitochondria, but not all have chloroplasts. What is the evolutionary explanation for this? So it is thought that the evolution of mitochondria event took place before over a billion years before chloroplast event took place. So this is the endoplasmic reticulum. This is the nucleus. This is the ancestor of the eukaryotic cell or the host cell. It is believed to have engulfed oxygen using prokaryote in order to be able to improve its energy, make more ATP molecules. And then the non-photosynthetic eukaryote then have in another event engulfed chloroplast. And so we have the development of photosynthetic eukaryote. Both the photosynthetic eukaryote and the non-photosynthetic eukaryote are proliferating and continue on Earth today. Now let's look at the cytoskeleton and cell surfaces. So the cytoskeleton, like we mentioned earlier, include microfilaments, intermediate filaments, and microtubules. Their functions include maintenance of cell shape, anchorage and movement of organelles, amoeboid movement, and muscle contraction. All of this very, very important. Checkpoint question: which component?
I cannot what?
So the cell's internal skeleton help to organize its structure and activities. So which component of the cytoskeleton is most important in holding the nucleus in place within an animal cell, guiding transport vesicles from the Golgi to the plasma membrane, contracting muscle cells? Again, please complete your checkpoint questions and email to me. So this is the three types, members of the cytoskeleton that have been identified today. Tubulin is a protein that builds microtubules. They can polymerize to get longer and depolymerize. So there are two types of molecules that make up the microtubules. Microtubules are very, very important for transportation within the cell. It's also during the cell division responsible for formation of the spindle fibers. So, so microtubules are important during cell division in that sense, helping to extend the cell as it polymerizes and gets longer, and then it depolymerizes to help to pull apart the chromosomes or chromatic sisters, as the case may be. The intermediate filaments, you can see them hugging the nucleus, helping to hold the nucleus in place, all these green filaments around the nucleus. And then the microfilaments made of actin, very, very important in maintaining the integrity structure. They're very close to the cell membranes, and they help in the movement of cells. Muscle cells use actin filaments and myosin for movement and so on and so forth. So originally identified from muscle cells, they are now known to be present in all cells. So in plant cells, if you remember, we said there's a plasmodesmata that allows cytoplasmic flow, content of cytoplasm to flow from cell to cell. Of course, the nucleus will not flow. It's bigger than any of the plasmodesmata. A pause that there are. And so let's do cytoplasmic stringing in Elodia. So you can see the flow of the cytoplasm going from cell to cell. Evidence of the presence of plasmodesmata are joining the cells of multicellular plants together, connecting them together rather. So scientists discovered the cytoskeleton using the tools of biochemistry and microscopy. In the 1940s, biochemists first isolated the proteins actin and myosin from muscle cells. And using the newly developed techniques of microscopy, electron microscopy, scientists established how filaments of actin and myosin interact in muscle cells during muscle contraction. And since then, scientists have begun working, and researchers have found actin filaments in all types of cells, single cells, multicellular cells, all of the time, essential for movement within the cell and movement outside of the cell to be able to move in its environment. So in the 1970s, scientists were able to visualize actin filaments using fluorescent tags in living organisms. The use of fluorescent tags and fluorescent microscopy in living cells has really enabled us to further expand our knowledge of the dynamic nature of the cell. And in the 1980s, biologists were able to record the changing architecture of the cytoskeleton, the sliding action of the muscle cell. So this is a cell. You see members of the cytoskeleton around the nucleus by the cell membrane. So in the cells of animals and a few sperm cells of plants, there are cilia and flagellum that help to move. These are made from microtubules. Cilia tend to be rather short and numerous on the organisms that have them. Flagellum tend to be very, very few, one or few in the organisms that have them. Structurally, they are similar. They are both made from tubulin. The microtubule arrangement in cilia and flagellum is referred to as a 9+2 arrangement, in which a pair of microtubules are in the center of a circular ring of 9 pairs of microtubules around. Flagella, they are longer than cilia, and they propel the cell from one to another by undulating boat-like motion. In some of the lab videos, you should be able to see the motion of a flagellum. You have seen in the previous videos of paramecium the action of cilia. The linings of our airways is filled with cilia. The lining of our lungs is filled with cilia, helping us clean and sweep junk into mucus that goes into our digestive system. Otherwise, the air we breathe in, which is often impure, would over time have allowed accumulation of dust and particles in our airways. Cilia work more like the oars of a boat, and the difference between flagellum and cilia are apparent, but they have a common structure, the 9+2 arrangement and mechanism of movement. So checkpoint question: person suffering from primary ciliary dyskinesia, also known as immotile ciliary syndrome, this is a fairly rare disease in which cilia and flagellum are lacking the other proteins dying. PCD is characterized by current respiratory tract infections, recurring respiratory tract infections, and immotile sperm. How would you explain this? Seemingly unrelated symptoms. Again, please complete your checkpoint questions and so these are scanning electron micrograph of cilia magnified 5,000 times. This is the flagellum in human sperm cell magnified 940 times. So you can see the single flagellum because so many cellular persons. This is a 9+2 arrangement. So this is a pair of central microtubules, and these are outer doublets. 1, 2, 3, 4, 5, 6, 7, 8, 9 doublets on the outside. The the central microtubules, they are linked together by cross-linked proteins and linked to the outer doublets by the cross-linking proteins. Along the length of the tubes, there are dyneins, motor proteins that are connected. Those are kind of like the legs of the flagellum. So when the flagellum is about to flap, one side remains stationary and the dynein, the motor proteins move on the one side, causing the movement, the typical movement of the flagellum to be seen.
Flagella, or singular flagellum, and cilia, or singular cilium, are microtubular structures that extend outside some cell and that assist in moving the cell's surroundings. Flagella are much longer than cilia. When a cell has cilia, their number is 4, whereas a cell will have few flagella or single flagella. Many protists have cilia. The sperm of many plants and animals have flagella. Flagella and cilia are assembled from tubulant subunits to form a circular arrangement of 9 pairs of microtubules attached to a central pair. Flagella and cilia bend to cause movement of either the cell or the surroundings. The bending uses energy derived from the hydrolysis of ATP.
So ciliary movement in paramecium. So ciliary motion. So, this is flagellum movement in Chlamydomonas. See the undulating motion. This is flagellum. See. So the extracellular matrix of animal cells functions in support and regulation. So animal cells, they tend to, we typically draw the cell membrane as the outermost layer, but they tend to secrete, especially in multicellular organisms, this elaborate extracellular matrix. It binds the tissue cells together and supports the plasma membrane, helps with communication with the cytoskeleton of the cell, between the cytoskeleton of the cell and the surroundings of the cell. So the ECM, it may attach to the cell through other glycoproteins that bind to membrane proteins. These proteins that are integral going through the phospholipid bilayer are called integrins. So integral proteins. So this is the extracellular matrix. So this glycoprotein context, this is collagen fiber, and these are integral proteins. Notice how they connect through their hydrophilic parts to the cytoskeleton, and then on the outside to the connecting glycoproteins. they are tied in the extracellular matrix. So in animals, collagen fiber, essential member, giving strength, especially in skin, muscle cells that needs to be connected together in multicellular organisms. So complete the checkpoint question, please, referring to figure 4.119. So which structures provide support to the plasma membrane? There are three types of cell junctions that are found in animal tissues. So these cell junctions help to join iron cells together. It enables interaction and communication between adjacent cells. So three types of cell junctions enable adjacent cells to adhere, interact, and communicate with one another. These specialized junctions are called tight junctions, anchoring junctions, and gap junctions. Tight junctions bind cells to form leak-proof sheets. Anchoring junctions help to strengthen, rivet the cells together, and gap junctions allow ions and small molecules to flow from cell to cell. So you have tight junctions in your alimentary canals that prevents the leakage of acid or the compositions of stomach or your digestive system from mixing with the rest of the body. Anchoring junctions hold your skin cells together, for instance. Gap junctions of the heart allows ions to flow, generating an electrical contractile force that enables the heart to pump and distribute small molecules from cell to cell rapidly. So checkpoint question, again, remember to complete and email them. A muscle tear injury will probably involve the rupture of which type of cell junction. So this is a tight junction. See, this fluid is coming in, but tight junction is there, it blocks it, it's just gonna go back. So the fluid is prevented from flowing across this layer of cells, which form the tight junction. It's an anchoring junction like Velcro. They kind of stick together and prevent tearing and falling apart of the cells. And this gap junction, look at the gaps in the ions can travel through.
Desmosomes, or anchoring junctions, appear as thickened patches in the cell membrane region between two cells. Desmosomes contain specialized proteins such as keratin, the same protein found in fingernails and hair, that increase the rigidity of tissues. Desmosomes, such as those found in epithelium, smooth muscle, and many other animal tissues, are button-like junctions that bind cells together and also function as anchors for fibers in the cytoskeleton. In gap junctions, or communicating junctions, two cells are separated by a small gap, which is bridged by specialized channels that allow water and small molecules. Gap junctions coordinate the activities of adjacent cells. For example, the hormone that stimulates one cell will often activate adjacent cells as well by the passage of intracellular signals through gap junctions.
Tight junctions, which bind cells together, are found predominantly in epithelial tissues, such as the lining of the intestines. Tight junctions form a barrier that prevents fluids from moving between the intestinal space and the space between cells.
So cell walls enclose and support plant cells. So a plant cell, not animal cells, has a rigid cell wall that protects it and provides skeletal support. It doesn't have the bony skeleton like animals, but it's able to stand upright and erect because of the rigid cell wall. Sometimes that wall is strengthened by lignification and as it ages and matures. So the cell wall of plants is primarily composed of cellulose, and plant cells have cell junctions called plasmodesmata. Plasmodesmata allow plant tissue to share water, nourishment, and chemical messages. Checkpoint question: which animal cell junction is analogous to plasma plasmodesma. So vacuole, this is plasmodesmata and singular plasmodesma. So through pectinization, respecting layer between this is the primary wall, this is the secondary wall, and this is the plasma membrane on the inside, and this is the cytosol on this side. This is the cytosol of the adjacent cell. So at the end of this chapter, you should be able to group eukaryotic cells structures into the 4 functions, according to the 4 functions. The structures that are involved in genetic control, structures that are involved in manufacturing, distribution and breakdown of materials, structures engaged in energy processing, and structures that provide structural support, movement, and intercellular communication. Checkpoint question: how do mitochondria, smooth AR, and the cytoskeleton all contribute to the contraction of muscle cells? So go through this table. It's just a review of everything that we discussed in the lecture. And if you have further questions, get back to me. So you should be able to describe the importance of microscopes in understanding cell structure and function, describe the 2 parts of cell theory, distinguish between the structure of prokaryotic and eukaryotic cells, explain how cell size is limited, describe the structure and functions of cell membranes, explain why compactization is important in eukaryotic cells, compare structures of a plant and animal cell, note the function of each cell part, compare the structures and functions of chloroplasts and mitochondria, describe the evidence that suggests that mitochondria and chloroplasts evolved by endosymbiosis. Compare the structures and functions of microfilaments, intermediate filaments, and microtubules. Relate the structure of cilia and flagella to their functions. Relate the structure of the extracellular matrix to its functions. Compare the structure and functions of tight junctions, anchoring junctions, and cap junctions. Relate the structure of plant cells and plasmodesmata to their functions. Describe the 4 functional categories of elements in eukaryotic cells. This table will remind you of the units of measurement. Remember what is called comparing size, looking at microscopic organisms relative to what you see with the naked eye, relative to what you can see with an electron microscope. So, thank you.